Hello, and welcome to this FastFacts presentation from Cell and Gene Therapy Insights and bioMérieux. I'm Avi Pinchbeck, editor of Cell and Gene Therapy Insights, and today we'll be looking at ultra-rapid microbial detection in cell and gene therapy products. Joining me today is Félix Montero-Julian, the scientific director for the Pharma Quality Control business of bioMérieux in Lyon. Félix has over 25 years of experience in industrial and clinical diagnostics and has been and continues to be extensively involved in the implementation and acceptance of rapid and alternative microbiological methods. If you would like to ask Félix a question about this FastFacts, simply use the Ask a Question box to the right of the viewing pane. And now, without further ado, here is Félix with his presentation.
So as you know, the sterility testing practice in cell and gene therapy is currently performed using cell growth-based methods that most of the time take 7 days to release the products. No technologies are available that can be analyzed samples directly to get results in less than 2-3 days, and competitive methods are still used in order to test the final cell and gene therapy products. So there is a fundamental need for rapid sterility testings in order to make available cell therapy products for patients faster. Because for many of these patients that takes benefits of these therapies, this is often the last line of treatments. They are very, very sick. In addition to that, innovators are focused on shortening the manufacturing process, moving from 10 to 17 days to 5 to 6 days to meet patient demands for life-saving therapies. And we are seeing that the faster manufacturing time that has been developed needs new quality control solutions that are fast enough to keep pace with faster manufacturing approaches. And in order to do that, we need to rethink how to control the sterility of these products and what new technologies can be used to meet these requirements. So one of the technologies existing in the market today is the scanner DI. And the scanner DI is a unique solution that combines speed and sensitivity, because this technology allows to be used and to test raw materials of bioburden, allows to perform in-process testing, but also testing on final products. And there are three main pillars here. The first one is that we can obtain results the same day. The scanner DI allows also to have a broad specificity capable to detect microbes, bacteria, yeast, and molds. And it's also a proven technology because it has been in the market for more than 20 years. The scanner DI is tested using what we call a ScanBio2 protocol, and is divided in 4 different steps. The first one is the filtration of the product to capture all the microorganisms present in the sample on the surface of a membrane. And then the filtration unit is the sample, and the microorganisms are labeled with a specific viability marker that will allow us to detect later during the scanning process using the scanner DI, the microorganisms that meet some level of requirements, some level of specifications that can be determined specifically by the algorithm that has been defined to be used by the scanner DI. And then the events that has been selected by the scanner DI, I confirm through a final validation using a microscope. So let me tell you how the system works now. So basically, the scanner DI is what we call a laser scanning cytometer. And we have a membrane that is roughly the surface of 21 millimeters by 21 millimeters. And you can see here on the left top of the slide, the laser scanning principles, in which the laser is handled by oscillating mirrors that allows to scan the entire surface of the membrane. And you can see here on the top at the right-hand side of the slide, a cartoon representing the different ways the microorganisms can be detected. So the distance between the different lines of the laser spotting the membrane is 2.2 microns, and the spot laser is 6 microns. And when the laser starts to run, like in the example here, you can see here the signal intensity recorded by the system, and this is increasing as soon as the center of the laser is going directly into the microorganisms. So based on these recording intensities, fluorescent intensities, is how we can discriminate between microorganisms versus spurious particles. So then the software can change, can determine what are the events that needs to be validated by the microscope. So the scanner DI technology is capable to perform ultra-rapid sterility testing in filtrable products, can handle volumes between 1 ml to 1 liter, can be used for final products and in process. The testing could be qualitative or quantitative. The time to results is less than 4 hours, and it detects contamination of single microorganisms, including viable but not culturable microorganisms here. It's also an all-grow-based method. It's what we call a direct method detection, and allows to run up to 25 samples per shift. And this technology, as mentioned before, is capable to detect bacteria, yeast, and molds, and has been already tested in more than 200 compatible products that ranging from antibiotics, isotonic solutions, chemotherapy drugs, hormones, anti-inflammatory molecules, anticoagulants, analgesics, diuretics, and vitamins. So today, in the next slides, I will show you what this will be the scanner DI next generation. Is the scanner DI cell burst for the detection of microorganisms in cell therapy products. So what are the main challenges of the cell and gene therapy products, and is this possible to perform an ultra-rapid sterility testing, considering that these products are not filterable? The volumes that are handled is very, very small, between 0.1 to 2 milliliters. This corresponds to the final products. The needs are to have a qualitative method, a time to results in less than 6 hours, and also capable to detect the contamination of single microorganisms, including viable but not culturable cells, continue to be an agro-based methods, and allows to still run to 25 samples per shift, and still having the capabilities to detect bacteria, yeast, and molds. And remember, in the case of the autologous manufacturing process, one batch is one patient, and many different cell therapy products are now in development, including CAR T cells, CAR NK cells, gene-modified therapies, mesenchymal stem cells, and others. So the ultra-rapid sterility testing for cell and gene therapy must meet at least these 4 requirements. Same-day results, 6 hours, less than 6 hours time to results. The method should be equivalent to the compendial method that is used today. Most of the time, these products are released after 7 days of incubation. Low testing volumes, 0.1 to 2 ml, and the protocol should be applicable to cell and gene therapy products. The detection of microorganisms in the products that contains a very high concentration of mammalian cells, ranking 10 to 6 to 10 to 7 cells per milliliter. So our protocol is called CellBurst and is dedicated for the cell and gene therapy samples. Basically, on the top of the ScanBio2 protocol, we developed a specific cell and gene therapy product preparation that consisted in lysis the human cells specifically without altering the viability of the microorganisms present in the sample. We have, because of the preparation process, two different membranes, one for anaerobic microorganisms and a second one for aerobic microorganisms. And after the apply of the cell burst protocol, the product is filtered, labeled, scan and confirm. And on the bottom of the slide, you can see exactly the same ScanBio2 protocol that has been used for filterable products. And in that case, the total time is 6 hours. So let me show you now the preliminary results for the applying the cells burst protocol here. So we use 3 different kind of cells. The first one is a cellular model that we use in R&D. Is the Jurkat cells that mimics T cells at a concentration ranging from 10 to 7 to 10 to 7 to 3 10 to 7 cells per ml. We use also lymphocytes, T cell lymphocytes, at the concentration of 10 to 7 cells per ml. And also we tested some final samples or CAR-T samples coming from a lab ProMaps at the concentration of 5, 10 to 6 cells per ml. And all tested cell types were compatible with the new cell burst protocol, and by definition, compatible with the scanner DI technology. In order to perform the preliminary limit of detection and equivalency study, we perform a study based on the use of the most probable number methods to assess the feasibility before entering into the verification and validation phases of this protocol. And this most probable number method is mentioned in different guidelines, including the PDA, Technical Report 33, and USP 1223 for the validation of LOD, performance and demonstration of equivalence between alternative methods and compendial methods. The protocol that was defined is as follows. So we use 3 serial tenfold dilutions of a microbial solution, including starting from 5 CFUs, 0.5 CFUs to 0.05 CFUs. We use 10 different strains, as described in the European Pharmacopoeia, chapter 2.627. As you can see here, 5 microorganisms corresponding to the aerobic conditions, 3 to the anaerobic conditions, and 1 yeast and 1 malt. And we run with 5 replicates for each method, scanner the and Petri dish or Petri cultures. And in that case, we use the Jurkat cells at 10 to 7 cells per ml. And the number of positives per method and inoculum is recorded to obtain the associated most probable number at its 95% confidence interval. So the MPN analysis compare 95% confidence interval between the different methods for different strains. And in both confidence intervals, the overlap and the detection limits are deemed not significantly different if these confidence intervals overlap. And another important parameter when you compare alternative rapid sterility tests versus the compendial methods is the testing of the non-inferiority. And a non-inferiority test based on 20% margin was performed on the observed proportion of positive samples for each method. And scanner DE is deemed non-inferior to the Petri dish if the lower limit of the 95% of confidence interval of the proportion difference is greater than less than 0.2 or less than 20%. And this hypothesis is that like described in the yellow box. So when you can see here the data, this is the MPN and the non-inferiority testing. And what we can see is that all the error bars of the different microorganisms here, each column corresponds to one strain. And the first error bars corresponds to the cell burst protocol, and the second one to the Petri dishes. And you can see here that all the error bars overlaps, meaning that the limit of detection are not significantly different. And the non-inferiority test based on 20% margin shows also that the scanner DI is not inferior for the scanner DI. So the methods are equivalent. In conclusion, the data presented here shows that the LOD is not different between the scanner DI and the data obtained using the Petri dish cultures. The scanner DI detection is not significantly inferior to Petri dish cultures, considering a 20% margin. And these conclusions of this data are very promising and confirm that the most probable number method is suitable for protocol comparisons, and therefore we will use is during the verification and validation of the protocol. So this is the initial results we wanted to share with you today. Stay tuned, follow us to be informed of the news and the advancement of the validation and verification steps of our cell-burst protocol, following the going to our websites listed here. And thank you very much for your attention.
Thank you very much, Felix, for a very interesting presentation. And thank you to our audience for watching today. If you'd like to download a poster version of today's talk, summarizing the main points, you can find that below the video. We hope you'll join us again soon for more Fast Facts.